Hi, my name is Elizabeth Corley. This is Ian, and this is Ari, and we will be presenting on behalf of Team MTB today. So MTB stands for Mycobacterium tuberculosis, and this is the virulent pathogenic bacteria that causes the disease tuberculosis. So just last month, Dr. Mario Raviglione, the tuberculosis director for the World Health Organization, said to BBC News, tuberculosis and HIV are now competing to be the #1 cause of death from infectious disease in the world. And so just last year, 1.5 million people died from tuberculosis. In fact, 1 in every 3 people are diagnosed with tuberculosis, and this is particularly true in developing and immunocompromised areas of the world. So why is it so infectious? It's infectious because of its mechanism of transmission, which is airborne. MTB enters the lungs, and the body responds with an immune response. So the body recruits macrophages to kill the bacteria. And usually this works, but MTB is a virulent bacteria, so it has special ways to evade this process. So as you'll see, so it evades this process, and macrophages and MTB start to proliferate within the tissues of the lungs and the lymph nodes, and this causes a granuloma. As this occurs, the tissues of the lungs and the lymph nodes begin to decay. So as horrible as this is, there have been many strides to fight against tuberculosis, and the 2 drugs used to TB are isoniazid and rifampicin. But there are 2 main setbacks with these drugs. So these drugs, they require long and strenuous treatment on the human body, which is not very effective. And currently, the epidemic is magnified by the emergence of multi-drug resistant strains of TB. And so these strains of TB do not respond to these drugs. So this renders the need for new drugs to treat tuberculosis. So Team MTB has made it its mission to do just this. We pose the following question: what established drugs can be repurposed to disrupt vital virulence pathways within Mycobacterium tuberculosis? So today we're going to take you through our methodology. We're going to tell you what we've done so far, where we're at in the process, and what we hope to accomplish in the next 2 years.
So we've established that Mycobacterium tuberculosis is a virulent organism. But what does this really mean? Well, virulence, or if an organism like Mycobacterium tuberculosis is virulent, that means it has the ability to infect other organisms and cause disease. It does this through a series of virulence factors that are created by different protein-protein interactions within the cell. One of the main questions we want to address with our research project is what happens if we remove or inhibit these protein-protein interactions. Well, we can see that upon removal of protein-protein interactions, we lose the virulence factors that are there, and we lose the virulence of the organism. And we call this attenuation of the bacteria. So our goal is to use an assay to screen a drug panel taken from the NIH of 446 different drugs to disrupt vital protein-protein interactions for 3 different pathways that we've identified. So these are the 3 different protein-protein pathways that we've chosen. We have the ESX1 pathway, which most notably contains the ESX-A and B proteins. We have the ESX5 pathway that has M and N proteins, and the FOP-FOR sensory system that has the FOP and FOR proteins. So the question you might have is, why do we choose these different pathways? Well, in one way or another, these pathways contribute to the virulence of Mycobacterium tuberculosis. For ESX1, A and B come together within the cell, they form a protein dimer, and once they're excreted from the Mycobacterium tuberculosis cell, they help Mycobacterium tuberculosis evade digestion from the organism that it is infecting. So here we can see a basic model or diagram of MTB. We can see 2 yellow proteins. This can symbolize A and B, M and N, or FOP or FOR. And what happens is they come together within the cell, and they form a protein dimer like that, and that contributes to the virulence of the organism in some way.
So now I'm going to go through the flow of methodology with you guys. First, we're going to establish a screening method that will determine the effect of each drug on each of the 3 protein interactions already just mentioned. At the same time, we're going to be preparing cells that contain each of the protein pairs for the MPFC assay. Once we have the cells prepared and the screen established, we're going to move forward with drug screening. And if we get any drugs that successfully inhibit the protein-protein interactions, then we'll move it on to further testing. So our drug screen is based on the MPFC assay. This is a method that tests whether or not two proteins interact. There are many methods that do this, but the MPFC assay is advantageous to us because it is performed in Mycobacterium smegmatis cells, or MSM. MSM is a bacterial species very similar to MTB, but they are non-virulent, so it's safer to work with. They are also faster growing, so we can cut down experiment time. But because they're very similar to MTB, they have a similar cellular environment, which allows us to minimize confounding variables that might affect protein-protein interactions, other than the drugs we are looking at. So during the MPFC assay, we're going to be growing these MSM cells in medium that contains an antibiotic called TRIM. So TRIM usually kills MSM cells. However, what we are doing is we're going to take each of our protein in a protein pair and fuse it with one out of the two fragments of the TRIM resistance protein. Now, when the TRIM resistance protein come together, they can allow the cell to gain resistance against TRIM and allow them to grow and live. So if our proteins of interest come together, they bring the fragments together, allowing cell growth. However, the fragments by themselves do not function. So if our proteins do not interact because of a drug inhibition, then the cells will die. Now we have the MPFC assay, but we need to adapt it so that it can analyze a huge number of drugs. We said 446, right? Yeah. So what we're going to do is we're going to incorporate high throughput screening, which is the idea of analyzing multiple samples at once. We're going to have a 96 well plate, well, multiple 96 well plates, and in each well, it's going to contain a sample of MSM with a protein interaction, and it's going to contain a drug. And because the MPFC assay reports if the proteins interact by cell growth, we're going to use Alamar blue. This is a dye that changes color based on based on cell growth. So we haven't confirmed this method with actual experimentation yet. This is just a pretty good theory, but we know what is going to happen. We can guess what's going to happen. If drugs interfere, then the cells will die because they won't gain resistance, and the Alamar blue will remain blue. However, if drugs do not interfere with protein-protein interactions, then the cells will grow and the Alamar blue will become more and more pink. So for the MPFC assay, we need MSM cells that synthesize both fusion proteins. So we're going to do this by making recombinant DNA, which is the first step of cloning. So recombinant DNA is a sequence that combines multiple strands of DNA. Here we're combining two strands. The top strand is a plasmid, a circular DNA, and we're going to use plasmids PUAV 300 and 400. And these plasmids actually lead to the synthesis of the TRIM resistance fragments. The other strand of DNA is the gene for the proteins of interest, which can be ESXA, ESXB, all those things. Here's an example with ESXA. So it produces the protein of interest. So when we combine the two DNA sequences, we're going to form the recombinant plasmid that leads to the synthesis of the fusion protein we need for the MPFC assay. Once we have those recombinant plasmids, we're going to put them into bacterial cells. So that is called transformation, and we're going to transform first into E. coli, which allows us to build up a stock of plasmids by growing these colonies. Then once we have enough recombinant plasmids, we're going to transform them into MSM, so we can use in the MPFC assay, so that these cells will produce both of the fusion proteins to test out the protein-protein interaction. We're going to have cells with ESXA and ESXB, ESXM and ESXN, and then FOP and FOR. So far, we've successfully transformed recombinant plasmids for ESXA and ESXB. And we can know this because the recombinant plasmids actually provide resistance against antibiotics other than TRIM. So we're going to grow them. We grew them on plates that have those antibiotics. And because they grew, we know that the recombinant plasmids are in the cells. We also took the recombinant plasmids out of those cells and we cut them back into their original pieces to check if we have the correct recombinant plasmids. And this is a gel electrophoresis method that displays each DNA sequence in bands. Here you see the plasmid PAV 300 and the gene for ESXA. And these columns show recombinant plasmids and what they include in it, and it includes both ESXA and POD 300, which shows success in the construct. Here's POD 400 and ESXB, and same success. So we brought those plasmids into sequencing so we can confirm if the sequences match with what we want, and these peaks confirm that as well. We may have these successes, but we've also had failure. You can see that column doesn't have ESXB, so the plasmid is not successful. We've actually had many failures. We have plates that don't have colonies grow. We've had all these gels that just don't have the bands we want. Like, seriously, what's wrong? What's wrong here? No, but it's... I'm just joking. It's all about refining our methodology and getting what we want, getting the results we need.
So to reiterate, we're currently in the process of establishing our screen and in the process of cloning. We have done it, as Ian showed, for A and B, but we still need to do it for M and N and FOP and FOR. Once these two things are completed, though, we can move on to the drug screen. The drug screen consists of this 96-well plate design, and we will be testing all of the drugs here. We'll also have space for our controls, both positive and negative, and other controls we're interested in testing for. And what we can see here on this 96-well plate is the Alamar blue will be changing in the wells, as Ian mentioned. And here, for example, in drug 1, we can see that all wells turn blue, and so the drug is inhibiting the protein-protein interactions. And in that case, we'd want to look at drug 1 for further testing.
So once we've found drugs that do successfully interfere with our interactions, we'd like to look at 3 aspects of those drugs. We'd like to look at the cell cytotoxicity of the drug, so whether the drug will do harm to a biological system and can be used. We'd like to do a compound structure analysis, seeing if structure will relate to function, to see if any other drugs will have similar function. And we'd like to look at the dose-response relationship to see a proper dose response that we can use in the human body.
Okay, so we've talked a lot about the research and all the lab work we're doing, but now I want to go into a bit about the social aspect of Team Gemstone. We've been affectionately called Team Best Friend because we're all great friends, both inside and outside of lab. We've also set up a pretty effective schedule for lab work. We actually require that each member go into lab for a minimum of 4 hours a week. And this basically allows each member to be independent and thoroughly understand our complete research methodology. So even though we are the best of friends, that's not to say that we haven't run into our fair share of challenges and problems. We specifically had some difficulties with the fact that even though we all go in for a set time into lab, that's not to say that everyone completes the equal amount of work. So we all started with different biology, different levels of biology understanding, and so sometimes things fail for some that wouldn't fail for others. We've also had trouble communicating daily results in lab and troubleshooting those failures afterwards, as seen by all the gels that Ian presented that didn't work. And lastly, we've begun to notice that this semester, when we started, all this lab work and the scheduling, that not every member is as fond of bench work as others. And so in the future, we look to allocate different parts of the research process according to everyone's interests.
So Ari touched on some of our challenges, but one of our greatest challenges was raising money for our research. Many of you Gemstone teams must know. So last spring, we successfully raised $5,000 with the Launch MD program. So this is one of our greatest successes. So to conclude, this semester, we've successfully cloned our first system, ESX1, into plasmids. In the next few weeks, we should be able to have we should have been able to clone our ESX5 system and our Popian R system. Next semester, we shall be testing all of the 3 drug interactions with our 96-well plate assay. So we'd like to extend our greatest thank yous to our mentor, Dr. Volker Birken, the Birken Lab for showing tons of support, our library, Jeremy, and of course the Gemstone staff. We also have some distinguished donors we'd love to appreciate today. Thank you. We'll take questions now.
So So you guys didn't have like a timeline or anything in your presentation, but could you share one? Like, I understand where you are in your research right now, but how long do you think that's going to take you to finish? And then how long are the next steps going to be? Oh,
So the question was, Clara wants to understand greater where we are in our timeline and how long it will take to finish the